Hi everybody, I hope Dan brought me a few minutes there. It's really great to be, it's an absolute pleasure to be a part of this group and a part of this project and this conference. So I've prefaced my talk with a statement about technology and stuff, and I hope it will become clearer as I give my talk why that's so important. There's a lot of smart science in this project, and the acoustic surveys that have been done, the bat detector work is for me really exciting, and it really opens up new avenues for understanding bat diversity. So I've done this work with the colleagues that you can see there. Does this also work like that? Right. So I want to just jump into some of the methods that we've used and then just give you a few snapshots of some of the results. So there are basically two parts of the survey. We've done actual capture surveys where we capture rodents and bats using standard Sherman traps, mist nets and harp traps, which some of you have probably seen before. A lot of the bats at least were released so that we could obtain release calls with bat detectors. So as to build up a reference library of bat calls, which is really, really important, particularly in a new unexplored area, to be able to make accurate identifications from bat detector recordings. And about 70 specimens were collected, voucher specimens of about 24 species that are deposited in the Durban Natural Science Museum and that have been identified by myself from detailed cranial and skin characters. The acoustic work, I think was somebody mentioned earlier that the number of observations, I think it was Steve, for this project was something like 13,200 observations, wildlife observations, which is unprecedented. Well, just from the bat calls alone, there's more observations. Each one of those calls that's identified is a bat pass. So an enormous volume of data that were collected from the bat detectors, from mostly these two models, a sonimeter and a bat. Sonimeters can allow passive recording, so you can install them. Batteries last up to about a week. So Goods and them had to carry a lot of weight of batteries on their trips and their transits, and I'm sure they cursed those batteries a few times. But they just run passively all night, and the bat is usually used for more active surveys and when we do release calls of bats and so forth. So this again is unprecedented, about 2,744 kilometers of river transects in which Goods was faithfully recording every night, or most nights when he could, recordings of bat passes. And altogether, in the different surveys between 2015 and 2017, more than 200 nights of recordings have been obtained. So the only tricky part about all of this is actually analyzing the data. And we did this two ways, manually. So myself, Dan, and Mark Keith from the University of Pretoria did manual classifications, and that in itself is a problem in trying to compare what Dan called X and I called Y, and then trying to correspond it. More recently, there's a lot of debate about using automated classifiers. This is really more easy to do for birds and frogs than for bats, but for many parts of the world, automatic classifiers are available, and we've made one for Angola, which at least can quite robustly identify bats to groups of species. But if you're wanting to know about the outliers and the rare species, many of that information gets lost when you use the automated approach. So we've tried to use both approaches, and we're still busy crunching data. That's what, by the way, a harp trap looks like with bats in it. If you've caught bats overnight, they fly into these fishing lines, two banks of a fishing line. They slip down into this trap bag, and they basically tuck themselves under some plastic there and go torpid. So it's a nice way of catching bats. I'm going to briefly mention another aspect of what Dan and I are doing. This is just collating historical records, starting with a GBIF search, which yielded 14,000 records. The really amazing thing, and I'm going to show it to you in a moment, I'm going to ask you a question, where's the hole? There is no hole with the mammal records. There's been recently a lot of new data added from 31 different institutional databases. 93% of the records come from two very reliable sources and very important sources. So the American Museum of Natural History, all their data, specimens that were gathered during Hill and Carter's Verne Lang expedition back in the 1940s, resulted in the book on the mammals of Angola. Really important work. Those records are now available publicly and well curated, and the taxonomy is up to date. Then also this institute in Portugal has consolidated all of Crawford Crabelle's, we've heard about him a few times, all his records, including the mammal records. So there's about nearly 9,000 records. Good records, solid, well-curated, taxonomically up to date. So this really makes this a useful resource for us, for mammals. And to be able to answer the question that Dan was asked earlier about how does the historical records compare with what we're finding. It's all there. So I'm still, and Dan was going to have to help me a lot, busy with the cleaning and cross-checking of this database, but I'm just going to show you a snapshot before I get into the bats and the small mammals, if I have time. This is what the record looks like when you plot the GBIF record. Where's the hole? So the skeptics will say, well, those records aren't very reliable, or they are just all large mammals, but I think I'll try to show you that they seem to be quite robust. Just to add other information that's available, this was the book, Bats of Southern Africa, by Ara Monogam, Woody, myself, and Corey Schoeman. This is where those records fall for the bats. And then Ara, myself, Woody, Christian Denis also brought out the bats of sub-Saharan, so the rodents of sub-Saharan Africa, and these show all the records of the rodents. You see mostly towards the western parts. Here there are big holes. Here you see the holes. Just to add to this, this is where new field captures were made in the 2016 season that I mentioned. Myself, with help of Goetz and Werner and others, and Sina Vaya and Mark Keith made collections in these areas, Kuchi, Gorge, and then the Quito Panavani areas. Then these are all the river transects that we've heard about already. So these are all points, as I said, where bat detector recordings were obtained. So again, it's, I think, unprecedented. Must be the largest acoustic bat transect ever undertaken. I'm quite sure that's safe to say that. And again, huge number of records. So just a quick snapshot. This is AMNH, should read Hill and Carter. This is the American Museum records I spoke of earlier. This represents the blue, the state of knowledge of how many species of different orders of mammals were known back in 1941. This brown shows all the GBIF records, and the green shows the two books by Ara Monogam, the 2010 Bat Book and the 2015 Rodent Book, just to give you some idea. And this, please don't hold me to that figure. We're still busy with the cleaning, but somewhere around about 280 something species of mammals from these databases. Look at that. Most of them are small mammals. So I want to dive into the small mammals. Now, as we've heard, bats are really valuable ecosystem. Well, we haven't heard, but I'm telling you, and you should know, bats are really important ecosystem service providers. And I don't really have time to go into it, but the pollination work, the dispersal, 95% of forest regrowth in tropical African equatorial forests come from seeds dropped by bats. I'm doing some work on bats as controllers, agricultural pest controllers in the macadamia industry. They save farmers about $50 million a year, 50 million rand a year in terms of the services they provide. And bats are also subject to a lot of recent discoveries, like some other groups, particularly reptiles and other vertebrate groups we've heard. So between 1989 and the present, we've added nearly 60 species of bats, African bats, from Africa and Madagascar. So you can see it's about 16% of the total that has been described recently, and you can see rates of description of species has increased in the last 2 decades. So I'm quite sure that with molecular work, we may find some of the populations from Angola are indeed cryptic species. So just to cut to the chase, 11 species were captured during this really short 2 periods in early and late 2016. And then 29 species were estimated from the bat detector recordings, bringing it to an estimated total of 33 species of bats. And I'm going to show you some pretty pictures because everybody's looking at pretty pictures, and this is National Geographic. So this is what some of the bats look like according to their families. I think I've still got 10 minutes. Okay. So, and I will get onto the rodents and shrews, I promise. Here's a slide or two for them. So the fruit bats, these are the 3 species we captured. There are 14 species recorded in Angola. This is a really special bat. It's a fruit bat that weighs 18 grams. Most of the fruit bats I'm used to dealing with weigh close to 100 grams. When I saw this guy in a net at the Quinavali source lake, my first thought was, that's a neonate that the mother must have been caught in the net and flown away. That's a newborn, and even for a newborn. And then it's actually the Anquitas fruit bat. It's a very interesting and rare, quite a rare species of bat. It's more found to the north and tropical Congo basin. It has whiskers like a cat and probably is important in pollination. This just shows the new record and the available records from Monaghan's book. So, coming to the leaf-nosed bats, only 4 species are recorded from Angola, a lot less than the diversity in West Africa, where you can have 10 species of herbicidarids at one site. But distinctive, based on distinctive sound calls, and on just one specimen, one pass, bat pass, we recorded it in this region, and this shows you its historical distribution. So these are range extensions, basically, but not that surprising. As Dan said earlier, there are not many instances of, there's only one in the case of bats where we're sure that it's a new country record. So again, we can go and look at other groups. These are what horseshoe bats look like. This is quite a common species in this Miombo woodland in the central highlands of Angola, is fumigatus, Rhinolophus fumigatus, this horseshoe bat. Again, distinctive vocalizations which make it possible to identify. These are imbanurid bats, and just based on acoustic recordings, we think we've got at least 2 species, one of which, Khedira ephra, is quite rare, but from these observations, from acoustic observations, it seems to be more common than we think. Again, based on our hypothesis that this call is indeed a Khedira ephra, we don't yet have any captured specimens of it to verify that, but it has quite a distinctive call. We caught no slit-faced bats. These are often called whispering bats because you can only detect them within a meter or so with a bat detector. And we never caught any of them, but there should be 5 species. With this large family of vesper bats, or evening bats, we captured 7 species and acoustically recorded 15. There should be about 23 species in Angola. One of them that was quite special is this Carivula argentata, a very attractive bat and quite rare. It's only 1 record previously, and it has very high frequency echolocation call, broadband call, so we recorded it just once. Another quite special bat is this endemic, the Leafnosed angolensis, long-eared bat, and we managed to get release calls, so which is very helpful. The only bat that was a new country record that we're sure of is this Pipistrellus rusticus. Some of the 2 or 3 other species of bats which we've obtained, which we've estimated the identification from acoustic recordings, would be new records, but sometimes the overlap in the echolocation parameters means we don't have that much confidence to be sure. Finally, the horseshoe bats, we only captured the free-tailed bats, we captured 1 species and recorded 8 acoustically. There should be 23 in the country. Quite a special one is this near-threatened species, Otonops martienseni, a very large, high-flying velocid or free-tailed bat, and it's quite specifically recorded in the lower reaches of Okavango into the Okavango here, although there's 2 records from further west. So quite a few range expansions and maybe 1 or 2 new records. This recent paper, it was a modeling exercise using a very large distribution database, and it shows that in Africa 2 things are important for bat diversity: highland areas or elevational heterogeneity and rivers. And so it shows the Angolan Highlands as being an important area with, it estimates around mid-30s, the species richness, and we get 33 species. So we seem to be getting to what the model says we should find. And if you could zoom in on that, you'd see that those sort of arteries of all the rivers are where the bat diversity should be higher along the rivers. It's also another recent paper by Dorothea Pio, and some of us showed that this Angolan area, particularly even in the future, by 2018, with climate change projections, will continue to be a hotspot of phylogenetic diversity for bats, the Angolan area generally. All right, I think I've got about 5 minutes, just quickly. I can't leave the rodents and shrews out. In fact, they're really more interesting in a sense, because we only captured 13 species, but when I sort of drew a polygon around the central highlands of Angola and extracted the data from GBIF, there should be 40 species, including 6 endemics. So we're not really scratching the surface with our rather limited Sherman trap expeditions that we've done. We needed a lot more Sherman trapping to pick up the really rare species. But fortunately, historical records give evidence that they were there, but we would like to know that those habitats still contain those rare and endemic species. The common dominant small mammals are this dormouse, that's mislabeled, sorry. that's shouldn't be mus, that's right. But that's a gerbil, and that's a striped mouse, These are the new species records I want to draw your attention to. For rodents, Sunkless virilla, there's no records previously of these dwarf shrews, which are more arid savanna type of species, often tied to the presence of termitaria. And then these two rodents, Zolotomus and Mus, both are fairly arid, semi-arid type species occurring in Botswana and Namibia. And these might be good examples of what we were talking about earlier, those arid fingers coming up the river valleys. And then these are just examples of the genera where we have the 6 endemic species: 2 Optimus, 2 Hylomiscus, sorry, 1 Hylomiscus, 2 Dendromus, and 1 Crossigurus shrew. So that's just an overall picture of the rodents. And in context, this just shows a paper that I published in 2015, which summarized terrestrial small mammal diversity in Afro-montane habitats across Africa. It's quite low in the Drakensberg, it's quite high in the Soutpansberg, where I work. And when you get to equatorial regions like the Rwenzori's, we get staggeringly high numbers of terrestrial small mammals. So we only caught 13, but as I said, we should be collecting around 42. So more work to be done with that, basically. And I think that's where I was going to end. I'm not going to go into too much detail about how we can use genetic data to do phylogenetic reconstructions, but since I have a minute, the Optimus that we collected in this study with Optimus ankytai, which is really what I think a bit of a missing link. So together with Woody and others, we've done we've worked a lot on radiation of the flay rats, the laminate-toothed rats in equatorial Africa. Their distribution corresponds, this is one of Woody's maps, with all these highland areas of Africa, and they allow us to reconstruct phylogenetic history, particularly this isolated West African population from the Cameroon volcanic line of 2 species of Optimus. How did they get there? Their closest relatives are East African. And then we don't know anything about at least 2 endemic species of Optimus occurring here. So again, the sequences are being done in the lab as we speak, and hopefully genetic data from these rodents and other bats are going to give us insights not only to undescribed cryptic species, but also to helping us basically unravel the evolutionary history and biogeography of some of these regions, and the sort of the drainage evolution stuff that Woody was talking about. So that's basically it. So thanks to the National Geographic and Wild Bird Trust and all these groups, and the Department of Science and Technology, the NRF that support the research chair that I occupy at University of Venda. Thanks very much.